So this is the next part, actually part 5 of our lecture here on chapter 43. So looking at the B cell immune response, we have an invader, a foreign antigen, present on the entire invader. So it's something present on the entire invader that is noticed by a B cell. And so the B cell binds to this antigen, and in turn, that causes the B cell to get activated and to start producing clone cells. And so those clone cells are going to now produce antibodies. So instead of just keeping the antigen receptor on the plasma membrane of the B cell, we have that B cell turning into a plasma cell that is releasing antibodies directly aimed at this foreign invader or this foreign antigen. And it's going to be using one of the ways that antibodies work at identifying the pathogen for destruction. And then a portion of those cloned cells are also going to maintain their receptors on their plasma membrane, and those are going to become the memory cells that are ready to respond to a second or third or fourth invasion by this same antigen. So one of the questions we evaluate is how do a vertebrate produce millions of antibody proteins if it only has a few hundred genes coding for those proteins? In reality, there are about 20,000 genes that code for millions of different antibodies. And so how that occurs is by rearranging the DNA during translation so that different combinations of receptors can be produced. So that is how we get this big variety. So we talked a little bit already about vaccinations, but the immune system, when it's exposed to a harmless version of a pathogen, you need to be aware that that means you're going to trigger an active immune response. So that what that means is that the immune system is going to be stimulated to produce antibodies to that invader, and then as a result of that as well, you're going to end up with some memory cells being produced, so that in the future we have a much quicker response to a second exposure to that antigen. And you can see that in this graph here, and we'll look at another graph a little bit later. And Jonas Salk is someone who developed the first vaccine against polio. And polio is caused by a virus that enters the body through the mouth and multiplies in the intestines and affects the nervous system, and actually can cause total paralysis in just a few hours. Also, 1 in 200 infections lead to permanent paralysis. So it's a really serious disease and was a huge success when the Salk vaccine was able to use an inactive poliovirus to develop immunity against this really awful disease. This is different from passive immunity, and we talked about this again a little bit during our case study, but passive immunity is going to arrive in two ways. It means that you're actually getting antibodies from another individual. Either you're going to get them from your mother through the placenta or through breast milk, or you're going to get a direct injection of antibodies that have been developed elsewhere. And both of these are only going to provide a very short-term immunity, but at least it gives you some protection. So the other part of our acquired immune response is our T cells. And the B cells were considered the humoral response. T cells are considered the cell-mediated response, because what T cells are responding to is an infected cell. So whether viruses or bacteria or parasites have entered the cell, the T cells are going to react to an antigen being presented on the surface of these infected cells. So the types of infected cells can either be the body's cells, which are showing excuse me, portions of an antigen, or they can be other leukocytes, particularly phagocytes, that have engulfed a pathogen and digested it and now are presenting a portion of that pathogen as an antigen on their surface. And there are two types of T cells. There's the helper T cell and the killer or cytotoxic T cell. So the way that a T cell is going to recognize the infection is that once a pathogen has been ingested by another cell, whether it's a leukocyte or if it's a body cell, what's going to happen is a portion of that antigen, a fragment, is going to be moved to a MHC protein located on the outside surface or the membrane of the infected cell. And infected, again, could be a macrophage that has actually just engulfed the pathogen. So MHC stands for major histocompatibility proteins. And again, these are proteins that are like receptors, essentially, on the outside of all body cells and including the leukocytes. So a T cell is going to know another cell is infected because of these MHC proteins showing the antigens on the outside. And this is a really great picture showing a T cell's antigen receptor. So these receptors are going around looking for these proteins that are being presented, these antigens being presented actually on top of the MHC proteins. So an infected cell or a leukocyte can act as what's called an antigen-presenting cell. And the T cell's response is similar to the B cell, where we have, in this case, the T cell is going to respond to a fragment of the original pathogen being displayed on an infected cell. And what's going to notice this is a helper T cell. So the helper T cell will bind to these antigens here, and then what they're going to do is then start this response process. So the helper T cell triggers both the humoral and the cell-mediated immune response, but the helper T cell themselves do not carry out those responses. What they do is they send out a signal which initiates the production of antibodies in the B cells, and that these antibodies are what are going to neutralize, as we saw before, the pathogens. They also send out signals that activate cytotoxic T cells to kill the infected cells. So there are two things needed for a helper T cell to activate this adaptive immune response. First, it needs a foreign molecule to be present that has bound to the antigen receptor of the T cell. And then next, that antigen has to be displayed on the surface of an antigen-presenting cell. And that antigen-presenting cell can be a dendritic cell, a macrophage, a B cell, or it could be a body cell. So we're going to have the same multiplication occurring once that happens. What we have, this is called a clonal response. So once we have that multiplication of a clone of other T cells, either the stimulation of B cells to make a clone, or the stimulation and differentiation of T cells into cytotoxic T cells or killer T cells. And the signaling molecule that's used is called interleukin-2, which communicates with the killer T cells and with the B cells. So what is the role of these killer T cells? Um, so when an attacker gets past the B cells antibodies in the blood and actually invades the interior of your cells, you need a way to get rid of those infected cells. And so that's where we get the killer T cells. So killer T cells, their job is to destroy an infected cell, and they do this by binding to that target cell and secreting perforin, a protein that degrades or punctures holes in the cell membrane of an infected cell. And in that way, we get apoptosis occurring. One thing, kind of a side note here, is that the blood groups are actually determined by the phenotype or the antigen that's present on the red blood cells. So our blood group, if you have an A blood group, that means you have the A antigen presenting on your red blood cell. Or if you're B, you have the B antigen presenting on your red blood cell. So you have an MHC protein that is presenting an antigen. If you're AB, you have both the A and the B antigens presenting on your cells. And if you're O, you have no antigens present. So the reason this is important is when we're getting transfusions. Because if you have this antigen on your A blood cells, and you put that A blood cell into someone with B blood, then this person is going to attack, this person's immune system is going to attack the antigen on the A blood and destroy it. So that's why if we have B blood or AB blood added, they have antigens that are going to cause their destruction, and they're marking by the immune system as something foreign. They have a foreign antigen. Likewise, if you have B blood, that means you're not going to attack anything with B antigens on it, and you're also not going to attack O, which is why O is the universal donor, because O has no antigens on the outside. So a person can get O blood or they can get B blood if they're a B blood type. But if you put A blood or AB blood, which also has A antigens on it, so both of these have the A antigen, that is an invader in the body of a person with B blood. Therefore, these blood cells will be destroyed, and that's the result is a clotting of that blood. So that's why you have to have blood matching and blood typing, so you know what type of antigens are present on the outside of a person's blood cells. This is another summary of the immune response. So we have a pathogen invading, and then we have antigen exposure. And the way that antigen gets exposed is when it's ingested by a macrophage or another type of phagocytic cell, like a basophil or a neutrophil. All of those are going to ingest the pathogen. And what then will happen is the helper T cells will see the antigen being presented on the outside of these and send out the interleukin message to signal the B cells and the cytotoxic T cells to then begin to get to work. The B cells will be involved in the humoral response. They'll go around looking for the pathogen, and once they come across it, then they will then start producing antibodies through the plasma B cells. And if the antigen, or the pathogen that has the antigen has actually already infected cells, then the T cells are going to send out their cytotoxic T cells to get rid of the infected cells with that antigen. And in both cases, both the B and the T cells are going to have a portion of their clones remain as memory cells for future attacks. This diagram is showing you the difference in response times. So this has the relationship of the memory cells and how they actually are going to decrease the response. So you can see here from the time of exposure of an antigen, in this case